It might be a question you haven't thought about in a while. Perhaps it came to you last as you gazed up at a clear night sky, blinking stars shimmering against an intricately and potentially infinitely illustrated canvas. Or maybe as you looked down into the shimmering waters of a local pond, each drop of water teeming with countless living organisms. It could even have been a question that you asked your friends at sleepovers when you were younger, watching sci-fi films on a Friday night. But today I want to pose the question, what is life? What separates it from the water in that pond? What's different about the leaves of a tree and the breeze that blows through them? And furthermore, how did life begin? And are we alone in the universe? These are some of the biggest questions in biology and in science. And to begin to answer, in this programme, I'm going to turn to evolutionary biologists, molecular ecologists, and even physicists who spend their days observing and characterising distant planets, searching for those that are Earth-like. Well, I'm Dr. Ben, and I'm very, very pleased to have you join us on this programme. We're going to start by talking to evolutionary biologists from the University of Exeter's Cornwall campus, Professor Sasha Dall and Professor Dave Hosken. So let's start by asking that question of the evolutionary biologists. What is life?
It's a very, very old question, right? And even, predates our understanding of evolution, predates genes, predates all the things that everybody kind of posits are, kind of central to life. As far as I'm aware, there are kind of two kind of main ways that people approach this. On the one hand, people talk about replication and the need and self-replication, the fact that what you're doing is, you can reproduce, and that reproductive lineages are at the heart of biodiversity. You can't understand biodiversity without understanding reproductive lineages and their history and their dynamics.
Replication with some fidelity, right?
You're talking about things that entities that can copy themselves, and they're not going to be able to copy themselves forever because nothing can escape the second law of thermodynamics. Eventually, it changes just because you can never have a perfect process in practice. So then the sweet spot is how long do you need to be faithful before it stops being a replicator, if you see what I mean. So, and people talk a little bit about that. And
it depends over which time steps. You look from time step to time step, there should be reasonably high fidelity, but over the vast tracts of time, the initiator to end point might be quite different.
Well, biological processes are defined. The relevant, important timescale for biological processes is the replication time. Is the unit, the time over which it takes to produce another copy, produce copies, right? So there isn't an absolute timescale. So for a bacterium, it's going to be much, much quicker. The world is a much quicker place than an elephant, right? So, this is why, when we work at an evolutionary scale and try to predict evolutionary dynamics, and we talk about time in them, we pretty much always have to be referring to sort of reproductive time, if you like, reproductive time scale. So yes, it's how many reproductive events are you keeping the kind of most of the structure intact. And in fact, you don't keep all of the structure intact in any replication process. Biology tends to be dominated by these information-rich molecules that allow for they're very stable from a biochemical perspective, from an entropic perspective. They allow the structure, the kind of core information, to persist for long enough for it to be considered a replicator and replication from, in an evolutionarily meaningful way. Some people suggest that you need this kind of replication to be going on for there to be life, okay? But then you have to include things like some types of crystals, some biochemical processes, autocatalytic networks, RNA, viruses.
So Professor Sasha Dall has highlighted the importance of having something capable of replication when we're talking about life. And we've just talked about viruses and whether these too fit that definition. I'm going to turn now to Professor Michael Sweet from the University of Derby, who's going to delve more into viruses and how to think about them in this context. And in doing so, we're going to move closer to some potential explanations for the origin of life.
That brings us on to viruses. So viruses are super cool. They're non-cellular, so they don't have cells like the single-celled organisms like protists and bacteria and things like that. But it's a non-cellular particle made up of genetic material and protein that can invade living cells. So they were first identified in 1898, and that was actually the tobacco mosaic virus, because it was causing mass problems for the tobacco industry. So a lot of people and a lot of money was put into that. And since then, thousands of viruses have been described, and quite a lot of them are disease associated. But the vast majority of them behave in a way that they need another organism to survive. Yeah. But even though we know so much about viruses now, the actual origins of viruses remains unknown. So not only do we not know the origin of life, but we don't understand the origin of where these viruses have actually come from. Have they come from plasmids? Have they evolved when the first bacteria evolved? Or did they evolve when eukaryotes came onto the scene? Or my likely theory is that they've evolved in many different instances, and hence why you get this plethora of diversity associated with viruses and the different types that you find. And so because of that, they are often referred to as organisms on the edge of life. And I find that a beautiful term, because life still finds a way. Life is still pushed in there. They still replicate. They're still passing on their genetic information, but they play a really interesting role in what's called horizontal gene transfer, and that's been associated with actual evolution of all organisms higher than the viral side of things. So basically every other aspect of life that we could discuss. So whether viruses are really vital as far as talking about life and whether we're going to find viruses on other planets or not is one argument. But then the importance associated with that and how it structures and changes other aspects of life is definitely something which can't be argued, really. And what you get with viruses is, again, these various different hypotheses, and I find these beautiful things to talk about. So one of the hypotheses for where viruses come from is called the regression hypothesis, and it's also known as the reduction hypothesis. And the argument here is that things like bacteria such as rickettsia or chlamydia can only reproduce inside host cells. So maybe over time, that these bacteria, which have that reliance on other organisms to actually reproduce, same as what viruses do in today's day and age, are just small cells which have parasitized larger cells and over time lost some of their genes, and therefore this idea of regression or reduction comes into play. And so that would argue that there had to be some other form of life to start off with, and then these viruses came in and were parasites after that concept. The next theory talks about the sort of cellular origin hypothesis, and this is also known as the escape hypothesis. Different people like to come up with different hypotheses which fit into this. And in my mind, this sort of echoes with the protocells, because this escape hypothesis talks about folded DNA and the actual aspect of plasmids, so pieces of DNA which can move between cells, or transposons, which are the mobile genetic elements, being able to move away from a cell like a bacteria and start to live on its own, and again produce a sort of parasitic aspect. So that's the escape hypothesis or cellular origin hypothesis. But again, that refers to the fact that you would have had a bacteria in this instance as the original origin of life. The next one is, and this is the sort of third and final, really, and this is the virus first hypothesis, or the co-evolution hypothesis. And this is the concept where they've actually evolved from complex molecules of proteins and nucleic acids at the same time as cells, and they have a dependency of cellular life for billions of years. So something which led to that, this hypothesis is the discovery of viroids. And viroids are similar to viruses. They're actually a classification of viruses, and they're molecules of RNA which lack the protein coat. So the protein coat is one of the essential aspects of what a virus is. There's all these things called virus-like particles, which are where you use things like transmission electron microscope to actually try and describe what viruses look like in anything, tissues and things like that. And because it's quite tricky to actually describe them, most scientists call them VLPs, virus-like particles. And but what we're looking for is distinct shapes and structures and things like that. And viroids are actually missing one aspect of what makes a virus. And interestingly, they do not code. So these viroids do not code for proteins and use the host machinery again for their replication. But it's another sort of missing link, which suggests that you might have had viroids first, then viruses, and then moving on to more higher complex organisms or even single-celled cells. So it's a fascinating aspect in those sort of contexts. And in fact, with these viroids comes other interesting aspects. So one of the things that people have looked at recently is you get certain viruses which are known as satellites. And these satellites, again, don't necessarily play by the rule. There's a good example. So this is a sort of a potential intermediate between viroids and viruses. So you've got viroids, satellites, and viruses now. And satellites obviously has connotations with space, so it's a beautiful aspect to talk about in this instance. But the hepatitis delta virus of humans has an RNA genome similar to viroids, but has a protein coat. And we know that viroids don't have protein coats, so it's not a viroid. And the protein coat is derived from the hep B virus, but it can't produce its own. So this suggests that you've got this halfway house where the hepatitis delta virus is a satellite, and it needs to be associated with the hep B virus, and it has to replicate, but it's not able to replicate without it. So you've now got this sort of co-habituation of viroids, satellites, and viruses, which really starts the building block of life, if you can imagine it in that sort of context. And if that wasn't enough, then you have to bring in this concept of prions. So prions are again a fascinating aspect, and they behave in a very similar way as viruses, but they're actually just misfolded protein. So it's an infection protein molecule that does not contain DNA or RNA, but behaves in a similar sort of context as far as disease causation, and causes things like scrappy in sheep and mad cow disease, which was brought to fame in the UK in the sort of late 90s, early 2000s, and then Creutzfeldt-Jakob disease, which is a virus which or a prion which causes quite severe degradation of the brain tissue in humans. And this discovery, so the idea or the concept of prions brings credence to the theory of the viral evolution from self-replicating molecules. So this escape hypothesis aspect. So there's lots of exciting evidence in all parts of biology and science, which tries to tackle this aspect of origin of life. And the thing what fascinates me the most is that you could quite easily get behind any of these sort of hypotheses, because they all have facts and figures associated with how they can be right. And I really like that, because science is, or in my book, science is all about chasing for that truth.
So far in our adventure into discovering what is life and trying to explain how it could have started, we've learned about the importance of replication. And in defining replication, we found that we had to consider viruses. And now, having delved into the topic deeper, we find that viruses could in fact actually explain the origin of life. As Professor Michael Sweet was explaining there, one potential scenario is that life began as self-replicating molecules which eventually became viruses, and potentially even through this system of viroids to satellites to viruses and eventually becoming cells. And even if this doesn't turn out to be the way that this worked, it's clear that viruses are absolutely essential for horizontal gene transfer, a mechanism that Michael Sweet was explaining there as being critical for evolution and life as we know it. But one key question remains in this story about how life could have begun, and that is the development of complexity of complex self-replicating molecules from seemingly nothing. So how can we imagine building up the complexity of these replicating units, and indeed of the vehicles that contain them, of the cells and so on? Well, it turns out there could be a reason linked to a fundamental law of physics, the second law of thermodynamics.
So there's one guy who argues from MIT who's arguing that actually something like adaptation, which is for just kind of, I guess, to get everybody on the right page with this, adaptation is the appearance of design, if you like, the appearance of
fit to your environment.
Yeah, fittedness, as I like to call it, fittedness to the environment. He talks about where you've got a kind of these gradients of exergy, high quality energy that's very usable, has a lot of work that you can do with it. It's not like a, because the second law of thermodynamics is telling you that you'll go from having, these kind of concentrated kind of high order energy sources to the energy doesn't disappear, it just spreads out and becomes less useful, becomes this kind of broad useless heat. So exergy is a term that I see a lot which seems to refer to this kind of the quantity of usable energy. But he posits that every time you get every time you get a source and there's a massive gradient in exergy locally, that you'll get kind of spontaneous self-organized things that will happen, like a lightning bolt. I mean, actually, he gives a nice analogy. He describes lightning as this: it's quite ordered, and what it does is it dissipates exergy at a high rate. It's really, really good at dissipating entropy. And he talks about life as being one of these processes that probably emerged as a way to dissipate, reacting to this fundamental. If it is true, if this view is accurate, that these kind of gradients create a kind of entropic pressure system. And so, but the things that emerge often represent the details of the environment in which that gradient is embedded. So the heterogeneities of the way that the energy can move through the system. So you do get some representation of the environment.
So high energy gradients could potentially, in theory, allow complex self-replicating molecules to emerge from seemingly nothing at all. Or at least from seemingly random background chemistry. But is there anywhere on Earth that there could have been such a gradient that could have kick-started life, that could have added that spark and turned background chemistry into complex self-replicating biological molecules? Well, it turns out the answer is yes. And understanding where to look could even tie in with the search for life on other planets.
So one of the first questions we need to talk about whenever we're looking at life is what exactly is it? And if you do a quick Google, you'll find that there are many different aspects associated with what life is and different people describing it. But by and large, you need some form of biogenic element. Usually carbon is obviously our life-based source on Earth, a source of energy. And most people think of light and photosynthesis in this region, some form of water, liquid water. And a lot of people argue that you need a suitable and reasonably stable environment for evolution to take place. So that all sounds perfectly sensible. And in fact, if you try and think of life in the general concept, it ticks all those boxes. But when we look further afield, we might not be looking for carbon-based life. We might be looking for something like silicon-based life or something associated with that. And in fact, silicon is obviously a major aspect, a major element, which is based in the deep sea. And some people have the theory that the deep sea is where life actually formulated.
So this idea about the origin of life based on energy and entropy, how does this relate to the deep sea environment mentioned by Professor Michael Sweet? Well, as it turns out, one example of these gradients, these energy gradients, where there's a lot of usable energy, so-called usable energy, in contrast to the typical energy we might think of when we talk about heat and temperature. Well, one of these gradients could have existed at deep sea vents, making the emergence of life there, however unlikely, favourable based on this idea of energy and entropy. Professor Michael Sweet, as an expert in aquatic biology, will talk a bit more about the possibility of the origin of life at these deep sea vents.
So the aspect of hydrothermal vents or deep sea vents and how life can be looked at is that it's the ideal conditions, or it's theorized to be the ideal conditions for the origin of life. And so this is why certain people are looking into that. It's a fascinating environment anyway, just on an ecological and a biodiversity aspect. But for the evolution chaps, this is really showing some quite exciting aspects. And what they found, so it's rather than sort of shallow pools and things like that, these deep sea vents are creating a hotbed, to give the pun, for creating protocells. And for protocells to be created, you need this hot, usually alkaline seawater associated with it. So, this idea or this concept of protocells, people have been trying to create protocells in lab environments for many years because these ideas of this protocell, and a protocell is a self-organized and ordered spherical collection of lipids. So it's thought to be the key stepping stone towards the origin of life. There's multiple competing theories associated with where and how life started, but these vents are among the most promising locations for life beginnings, and new findings which have been published in places like Nature, Ecology and Evolution are leading aspects of information onto this sort of concept. So it's these areas are mineral-rich chimneys, and they have that alkaline aspect with acidic fluids, and it's providing huge amounts of energy, which is, as we remember, one of the important aspects associated with life. But interestingly, down in the depths, obviously, you're not talking about things like photosynthesis, and in fact, quite often you're not talking about things like heterotrophic feeding, so actual food. You're looking at things like chemosynthesis, so the facilitating chemical reactions, particularly between things like hydrogen and carbon dioxide, to form these increasingly complex organic compounds. So people are moving forward onto this, and I believe, and I need to check this, but I believe that protocell formation has actually been formed in a lab now, sort of again leading credence onto the fact that this is actually a sensible origin story.
So we've spoken about replication as a defining feature of life. We've visited potential early life and the potential for viruses not just to be considered as on the edge of this definition, on the edge of life, but in fact potentially explaining the origin of life. And now we visited the deep sea to hydrothermal vents, which could have represented one of the places that could have birthed early life. A theory that could potentially also be made even more likely given ideas about entropy and energy and usable energy gradients based on fundamental laws of physics. But where do we go now in this discussion? We've provided just a few perspectives on what life is and how we define it, and potentially where and how and why it began. But it leaves us with perhaps the biggest question. Are we alone in the universe? And for this, I'm going to visit the University of Exeter's Streatham campus. I'm going to talk to astrophysicist Dr. Raphaëlle Haywood, who is a leading expert on identifying exoplanets that are potentially Earth-like in nature. So come with me as we now travel to Exeter to continue the programme. Well, I've just arrived on the University of Exeter's Streatham campus. About to pop into the physics building, towering over the rest of the campus. Just like the researchers inside, I guess it reaches for the stars. So I started the interview by asking Dr. Raphaëlle Haywood whether she believes that there may be other Earth-like planets out there.
You're asking the big question there. I mean, for me, as an astrophysicist, I did my PhD on looking for exoplanets. I'm really passionate about. I mean, I think it's amazing that we can, learn about these worlds that orbit stars that you might not even be able to see in the night sky. It's the precision of our instruments is just incredible, and our ingenuity in analyzing the data. And yet at the same time, the more I find out about these planets, in particular those ones that are in the Goldilocks zone, and we're always striving to find ones that are going to be more similar to Earth, the more I look, the more I realize, actually, no, we're not going to find something that's like Earth. It would be like saying, okay, so we know now today that, 1 in 5 stars like the Sun has a planet that is roughly the same size as Earth and roughly in the Goldilocks zone, basically. And 1 in 5 stars, so there's about 300 billion stars in our galaxy that are roughly like the Sun. So you're talking about 60 billion planets that are temperate and Earth-sized, roughly. But does that make a planet Earth-like? Does that mean it's going to be like Earth? I mean, it's like talking about, you look at, we're at 7 or 8 billion people on the planet here. Every human is unique, though, right? I mean, we're all human, but even twins are all going to have different. They have different character. They have different personalities. They have different experiences in their lives, and so even identical twins are never going to be quite the same. And when we look for these planets that we want them to be similar to Earth, I think it's going to be the same thing. We're never going to find quite the same thing because we need something that's really, really finely tuned. And the reason for that, I think that's the crux of the matter, actually. The reason we need something so finely tuned as our own Earth is because so by we I mean humanity, but I mean more generally life on Earth, we evolved with our planet for the last 2, almost 3 billion years. And so we made it what it is today.
And next, I wanted to ask Dr. Haywood about what I understand to be a really key barrier to ongoing research in locating potentially Earth-like planets. And I believe that one of the big problems is actually background noise or interference on the tools used to find them.
Yes, that's one of the things that really fires my passion for astrophysics, because we're looking at things in such detail that currently the main obstacle to find planets that are temperate, so the same in a 1-year orbit around a sun-like star and about 1 Earth mass, even planets bigger than that, to be honest, but looking for these small rocky planets, the main challenge is actually it comes from the stars themselves. So when we're looking for planets beyond our solar system, we're always actually looking at the impact that the planet has on its host star. So remember, I was talking about the Doppler method. You've got the planet is pulling on the star, and from that we can deduce that the planet's there and how massive the planet is. The thing is that when you're looking at these signals, they're so small that you have to take into account the fact that the star is not a constant light bulb. The star is like a big, massive ball of gas and plasma, and you've got flows coming up and down, and then you've got the flows interact with the super strong magnetic fields, and you get things like on the sun, you've got these sunspots, you know, those dark areas of super strong magnetic fields, and they inhibit the convection, the bubbling up that's happening on the surface of the sun. And stars all have that, and all of this dynamic, ever-changing movement creates changes in the color of the light. And so remember how I was saying that you measure the Doppler shift by looking at, you know, the star is moving back and forth in your line of sight, and it creates blue and red shifts. Like the light goes blue, red. Well, all the flows on the stars, they create little, you know, when you've got like a massive thing coming to the front, rising up to the surface of the star, it looks like something's coming towards you, right? And then it sinks back and it looks like it's going away, and so you've got blue, reds, and you've got all these micro signals that just sometimes completely obscure your like signal coming from the planet going around the star. And it's incredible that we're even able to say this.
Yeah, I think it's probably going to surprise me. I think one thing that you said in that explanation there, which was awesome, so thank you for that. But one thing you were saying was about, you know, it almost sounds like, you know, we've got the tools and that's totally fine. You know, we're ready to see this unbelievable sort of resolution. Mm-hmm. But actually, what do you do with the data that's coming out of that? And it sounds like that might be one of the key hurdles actually that you're trying to get through. But for example, this noise. I mean, is this one of the things is this like a really key stepping stone to sort of being much better at doing this, basically?
Yeah, absolutely. And before I moved to Exeter as a lecturer, I was in the States as a postdoc, and I worked as part of a committee of experts who advised NASA on building a roadmap for the next decades for finding rocky terrestrial planets. And one of the main outcomes of our reports was that we need to understand basically the physics of what's going on the surfaces of stars. We need to be able to model the stellar variations, the stellar variability, that intrinsic variability that stars have, that is driven by fluid dynamics and magnetic fields, basically, and the interplay between the two. And that's key. And if NASA wants to find, you know, terrestrial temperate planets, it needs to understand the stars that they orbit, NASA and anybody else.
It's fantastic. I think, for sure. I mean, I'm really hoping actually anyone listening in will think, wow, that's just absolutely remarkable that you can do this.
And to add to that, you know, just to be clear, the solution to that problem is not going to be to build bigger and better telescopes or to go to space to escape, you know, the blurring atmosphere that is around Earth that blurs our, you know, it makes stars twinkle and it pollutes our observations as well. But the solution to this is more brain power, is to actually understand, you know, the mysteries of the stellar interiors and surfaces.
So presumably once you sort of start to get a handle on that and you can start to build sort of, what would you do? Could you kind of build sort of models that sort of account for that, and then with the data you have you can sort of remove the noise almost? Is that sort of the goal?
That's the idea, absolutely. And we can do that. Actually, I've been working quite a bit. Part of my PhD was on this. You can look at our own sun, because the sun is the only star that you can resolve at high spatial resolution. You know, you can see the features on it. And at the same time, what we've been doing, the collaboration that I'm part of, we've been observing the sun as if it were a distant star, and we've been observing it with the same instrument, the same spectrograph that we use to hunt for planets. So we do a lot of, this is the HARPS North collaboration, the High Accuracy Radio Velocity Planet Searcher in the Northern Hemisphere, and it's this instrument on this telescope in La Palma in Spain, and we built it to follow up planet candidates that were discovered originally by the Kepler mission back in the 2010s. And since then, you know, we had Kepler, and now we've got, then Kepler broke and it became K2. And now there is the TESS mission that's finding a lot of planets in our celestial neighborhood, as in around the brightest stars in our neighborhood.
So multiple ongoing projects seek to further characterise and find exoplanets, particularly those that could be Earth-like and therefore could potentially harbour life. And that's where you come in. Dr. Haywood told me about initiatives that mean everybody can get involved in the search for exoplanets.
You can become an exoplanet hunter yourself.
Now we're talking.
Yes. So I think it's led by Oxford. There is a website that has been built by astronomers, astrophysicists, and of course people who know how to build slick websites. And they've basically dumped all the data from Kepler, and I'm pretty sure there's test data on there as well, and K2. All this data is publicly available from NASA. And on this website, they've put the reduced data, and basically you can go yourself, and they train you on how to eyeball these light curves. So the TESS and Kepler missions, they look for transits. So they look for little dips in light, periodic dips in the brightness of the star as the planet goes around the star, and they train you, and then you can go out and eyeball, however many light curves you want to eyeball. You want to take regular breaks.
That sounds like it comes from experience.
Yes. I'm pretty sure some of my eyesight prescription came from after a session of eyeballing, an intense weekend session. But yeah, and you can basically, it will show you what the algorithm thinks are transits, but you have to look at it and you can say, what you think it is. And this is incredibly useful because the fact is that we're getting so much data. We're talking about hundreds of thousands of light curves, millions of light curves, millions of potential transit events, and AI is not able to say for sure what it is, and only a human eye can do that. A human eye and a human brain, the two connected. So actually there have been several discoveries of planets like this, planets being confirmed by teams of citizens, and people have published papers with astronomers, and often they're actually really interesting planetary systems, because there's stuff that the algorithm hasn't picked up, and it's not going to be something super obvious, because the super obvious stuff gets picked up often by the astronomers themselves. But yeah, we need people to eyeball these light curves.
Yeah, that's awesome. To me that comes right back to something you were saying a bit earlier, actually, about one of the key hurdles that we need to get through is actually just brain space and brain power, and actually that comes back to that a bit to me in terms of to make these next steps. It all sounds really fabulous, but actually it sounds like we've just got to do the work. Actually, we've got the data, just need to do the work, and anyone who's available to help do that, then brilliant. We can all sort of be involved together, which is awesome. So returning to a key point raised by Dr. Haywood to close this section of the programme. She told us that there is not likely to be another Earth or another planet out there that is very similar to Earth. She explains more about this in some published work.
If you're keen to find out more about why there's no planet B, my collaborator and my collaborator Arwen Nicholson, who's a postdoc here in astrophysics at Exeter as well, we recently published a piece called There Is No Planet B in Aeon, which, if you're going to read it, I recommend it, and I really recommend that you read the comments as well and leave a comment, because clearly this is a topic that really it struck a nerve. People have very strong feelings about it, so I'd love to hear how you feel about it as well, and continue the discussion on it.
But we still know so little about our life on Earth. We know very little about life in the deep, and obviously we know even less about life up in space.
The more I find out about these planets, we're always striving to find ones that are going to be more similar to Earth. The more I look, the more I realize, actually, no, we're not going to find something that's like Earth.
Well, thank you very much to the special guests who contributed to this programme. Thank you to the University of Exeter's Professor Sasha Dall and Professor David Hosken. Thank you very much to the University of Derby's Professor Michael Sweet. And thank you to the University of Exeter's Dr. Raphaëlle Haywood. You also heard some music produced by yours truly, and also a fantastic track earlier on by Dave Buckley and the Watchwood. That's called Still She Sleeps. Hope you enjoyed the programme. Goodbye for now.